Ugo Basile. But this is how we started probably, I would say, 8 years ago, with a simple proof of principle. So this is a small ring where you see two soldering units, and here a system of tubes that are connected to a cold pump. And we actually wanted to have a system that allows us to have mice deliberately decide where they want to go without being attracted to corners. And after exposed 1 hour to the different floor temperatures, the mice actually sitting in their preferred floor temperature. And after showing this proof of principle, we have actually made a larger design, and we make temperatures more stable with two Peltier plates. We adjusted the temperatures between 15 and 40 degrees for a start, because we simply wanted the mice to choose non-noxious temperatures. They will certainly be repelled by the 15 degrees and by the 40 degrees, but there is no noxious experience. So they are free to decide the direction they are going and to choose the temperature that they feel is just right, not too hot, not too cold, and find the floor spot where they have the highest level of satisfaction. This was our initial setup. We were in a large room. We had two custom designed setups, and we used cameras, normal CCD cameras from Logitech, and we placed a lid on top of our ring to make a distribution of zones that can be recognized by a software later. And we developed a custom designed software that analyzed both the entries to every zone, so how often the mouse goes in any of those zones, as well as the time and seconds that they spend in each zone. So you can see that the symmetric design of the ring, that means every temperature is represented twice, and you have one large zone, which is the cold zone, and that allows you to make sure that your software always takes the larger cold zone as a reference and applies the right temperatures to the results. We validated this system by using sensory transducers and transgenic mice that are lacking the sensory transducer, the cold temperatures. Pretty much everybody of you knows about the TRPM8 receptor and the TRPA1 receptor and their importance in the peripheral nervous system. We used the knockouts, so we placed the C57 Black 6 wild type TRPM8 knockouts and a double knockout of TRPA1 and TRPM8 into this system. And we used first the 1540 paradigm, and in the first 30 minutes you see that there's a mild avoidance of the C57 Black 6 mice towards warmer temperatures, so they are dragged. The red line is the random line, so you also see that at the beginning of this exposure, the double knockouts, they are behaving completely random, different from the TRPM8 knockouts and the C57 Black 6 knockouts. So they are spending 50% of the time in the middle of the ring, and they're not displaying any temperature preference. If you expose them for a longer time, so for at least 45 to 60 minutes, you see that all of these strains develop a certain preferred temperature. The C57 Black 6, they are still avoiding the cold temperatures very persistently, and you also see a mild, stronger avoidance of the TRPM8 knockouts. That was recently also described by Gary Lewin in a paper about worm perception. And you see that the double knockouts, they are developing also a strict avoidance of the cold temperature, but in this case it develops over a longer time, so you can actually recognize that the temperature preference assay can very precisely differentiate between these phenotypes. Then we applied a cold stress to the animals, and we applied 10 degrees less, placed a 530 environment, and you can see that in this environment, in the early 30 minutes, the double knockouts are also starting to prefer warmer temperatures. So it's simply you can differentiate between those two environments, how the double knockouts have a precisely different phenotype from the single knockout and the C57 Black 6 knockout. And if you leave them there over time, you will see how this phenotype develops over time. So there's definitely more about temperature sensation to be discovered in the future. So it's not all about TRPA1 and TRPM8s, as you can see, that these double knockouts are still avoiding the very cold temperatures pretty efficiently. So here are a couple of basic facts about the temperature gradient ring. It consists of a hot and a cold plate. Placed on top is a aluminium circular ring. Inside you have this transparent wall. And outside you have a invisible ring made of steel. On top you have in the center a wide angle camera that is able to grasp the entire area, and you have 4 different illuminations that give you a very solid illumination of the entire ring, and the mouse will not be able to find any cues because everything is evenly illuminated. Furthermore, you have insulation, so very solid insulation made from some Teflon material, I think, and this allows you to place this in a normal room at room temperature, and it will still allow the gradient to be established across the entire ring surface. So we have even floor temperatures, gradient floor temperatures in a normal laboratory environment. This system is connected to the Animaze software, which allows the video tracking of the mouse. And the Animaze software is pre-configured to plug the TGR in, so it's a plug-and-play system. And it recognizes the mouse and as well as assigns the correct temperatures to the field. So it has every plate has a temperature sensor, and between on one side you have in 1/3 each a temperature sensor, and from there it infers the temperature for every of those virtual zones. The basic benefits towards other systems are actually that there are no bias of these animals attracted to corners. There are no visual cues that irritate the eyes. And you can also use infrared illumination for tracking in dark conditions. You can do this without a looming experimenter. You can place this in a room where the mice are fully by themselves, and return after 60 minutes, clean the setup and put a new mouse. So you will completely eliminate your experimental bias. The symmetric design is also of advantage because it will allow you to represent every temperature twice and give you more accurate results, and at the same time the mouse has more freedom to move in a larger area that it can move in. So I wanted to show you a couple of results in the past. A knockout that we have characterized is a TRPC5 knockout mouse. We found that this TRPC5 receptor is cold sensitive, and at the time we did some skin nerve recordings. We recorded nociceptors, and we actually found that the TRPC5 knockouts, they have much larger responses in the cold sensitive nociceptors in the periphery. And here's a statistical view of the responses. These are stimulations of the receptive field with cold temperature, and the TRPC5 knockouts, you see they have like 4 times larger action potentials to cold stimulation. So we assume that they may also be more sensitive to cold, and we tried to characterize them in a 2-temperature choice assay at the time. 2-temperature choice assays were quite fashionable, and they were recently developed for the characterization of the TRPM8 knockout by David Julius and Adam Pataputian's lab, and we used the same design. So we use pairs of temperature, 30 versus 30, 27 versus 30, and so on. And we place the mice always on the colder temperature to give it a drive to move. But as you can see, we did not find a phenotype, so there were no differences. But the TRPM8 knockouts, they have a strong difference, though they don't distinguish temperatures up to 15 degrees. So despite this very large cold gain of function in the peripheral nociceptors in the two temperature choice assay, in the past we did not identify any phenotype, but with the TGR we actually found a strong cold hypersensitivity phenotype. This is actually a very nice way to plot it. You can also plot not only plot a cumulative display of the preference temperature, but you can also make a cumulative distance with the animes. And here you can see the different zones, 15 to 40 degrees. I just inferred the relative temperatures, and you see the cumulative distance actually in meters that the mice are covering. The wild-type mice are here, given in blue, and you see the cumulative distance from the colder zones, and you see that the wild-type mice are centering around 31 degrees, while the TRPC5 knockout mice are centering about 3 to 4 degrees warmer. So the cumulative distance plot, which is also included in the Animaze software, is a very nice and sensitive way to find differences in phenotype. We are also making this is a new research which we haven't published yet. We are exposing our mice in the cold chamber at 4 degrees for a month. They're getting a nice nest material to make and an igloo where they can hide, and they're always two in a cage, so that's not very cruel for the mice. They adjust to this change in temperature, and we were also able to find a very nice and interesting phenotype. When the mice are placed for a month at 4 degrees, they're actually becoming more warm-liking, so they are preferring the warm temperatures. And we find a similar change in the cumulative distance plots shifted to warmer temperatures by around 3 to 4 degrees. So other experiments we are currently doing is we're looking for the role of tail vasomotor response in temperature choice behavior. We're looking at ADRA2B knockout mice. I think this study will be published next year. It's almost done. And we're also looking at the role of the brown adipose tissue function in our temperature choice behavior. And the interesting thing is that the brown adipose tissue does not contribute a lot to temperature choice, so in contrast to the tail vasomotor response, which seems to contribute a fair bit about temperature choice behavior. There are a couple of recent publications with this device which I can recommend you. So Makoto Tominaga has extensively worked with this device, and he looked at pretty much all the TRP knockouts: TRPA1, TRPM2, TRPM8, TRPV1, TRPV3, and TRPV4. And he did present a pretty thorough analysis. He also plotted the body temperature when the mice were in the temperature gradient ring. So you can see that mice are getting a bit excited when they're getting in the temperature gradient ring. Body temperature rises. That's the subcutaneous temperature and the core body temperature. So, for example, if you're using UID devices subcutaneous to implant, you have a pretty good imagination of the core body temperature, because that seems to be tightly correlated. So this is a simple way that you could combine with the temperature gradient ring. So Makoto, in his paper, he analyzed the spend time per every zone, the travel distance, the moving speed, and the acceleration. So this paper gives you actually a pretty broad view of what is possible with the Animaze software in this device. There's another recent publication from Makoto Tominaga, and he used this for a model of peripheral neuropathy in diabetes. And he looked also at certain TRP channel knockouts, and what you can see here is spent time given versus zone temperature, and you see a clear shift in his diabetes models to warmer temperatures, which is absent in the TRPV1 knockouts. So he also, this is approximately also a 3 to 4 degree difference, and I think he could also use the cumulative distance plot to plot this probably a bit more consistently. And this final publication is from Irmgard TGR, who I know pretty well. She's in Frankfurt University, and she works with knockout mouse models. And recently she published a very nice paper about the function of nucleoredoxin, and she used nestin Cree mice as well as advilin Cree mice to compare the pan-neuronal function of the nucleoredoxin as well as the sensory neuron function of nucleoredoxin, and she also found a very nice phenotype, avoidance of cold temperatures towards heat nociception. So these are the most recent publication with this work. Here are some more references, including our 2 papers and the ones that I've just talked about. This is from a group in Naples, and they use this mainly for pharmacologic phenotyping, so there's not a lot of data from the TGR. It's not mainly a pharmacological paper, but they also used it for testing some compounds. So that's